Okay, hi everyone, welcome. My name's Catherine, and I'm going to talk a little bit about RNA-seq analysis. So on Thursday, before your group projects start, you will have two exercises to do, which both use RNA-seq data. You will have already looked at some RNA-seq data earlier in the course, but these ones just dig into it a little bit more. So before you do those exercises, I just wanted to go over some basics about what RNA-seq data actually is and how we generate it. Okay, so when we talk about RNA-seq, what we're talking about is sequencing the transcriptome of your organism, your sample. So sequencing all of the things that are being transcribed in a pool of cells. Why do we do it? There's a couple of different reasons for doing this. When I ask my MSc students this, they usually come out with ideas which are in this sort of second bullet point bracket. So they might talk about understanding what's different between different cell types, understanding what's different between different life stages, understanding what's different over a time course, understanding what's different if you grow your organism in different media or put it or at different temperatures or in slightly different environments. The second bullet point is what we generically call differential expression. Basically, the idea of asking, if I change something, how does the transcription change in my system? The third bullet point here is basically just taking that idea a little bit further. So rather than just looking at two different samples and comparing them directly, it's more about looking at what genes change together over a broader scope of conditions, if you like. And that can help you to understand, for example, which genes might be regulated by the same promoter or part of the same promoter cascade, transcription factor cascade. The first bullet point I've actually put at the top because I think it's one of the probably the most important uses of RNA-seq data, actually, and it's almost always overlooked. It's something that certainly my MSc students almost never come up with on their own. But actually, it's critical. RNA-seq data is critical in understanding gene structure. And understanding gene structure is critical because if you have the gene structure wrong, then your protein prediction is going to be wrong. And if your protein prediction is wrong, then things like determining what your gene does gets much harder because you can't look at things like PFAM domains or orthology or any of those things. Okay. So what we're talking about here, this is an overview of what we're talking about here. The pink box at the top is representing what is happening in vivo in your system. So you're going to start with DNA, and I'm sure everyone is aware of this molecular biology, but just to be clear that we're all on the same page, you start with DNA in the genome of your organism. At least for everyone in this room, maybe not if you're working on certain viruses, but that's an aside. And your DNA encodes genes. And when a gene is transcribed, the DNA for that gene is transcribed into pre-mRNA, which basically mirrors what's in the genome. But that is not that's not the end of it. When we in order to make mature mRNA that can then be translated into proteins, we need to splice out the introns, which are present in our genomic DNA. And what we end up with is a mature mRNA. It has a cap on one end, it has a poly-A tail on the other end, and the introns are gone. And that will become important later when we talk about gene model prediction from RNA-seq. So the mature mRNA is what we're going to extract from our samples. We'll usually use a poly-A selection mechanism of some kind to make sure that we're only getting the mRNA and not anything else. And we're going to fragment that. So we're going to turn it into lots of small fragments. We'll transcribe them. So what we're actually going to sequence is lots of double-stranded cDNA fragments that have been reverse transcribed from the RNA. So they're representative of the RNA that was in your system. And then we sequence those using a high throughput sequencing system, and it's most common here to use Illumina for this. That gives you lots of fragments of cDNA, which have obviously come from your RNA. And the next thing we do with those is we align them to a reference genome. So here we've got a reference genome here, and all of these lines above it represent individual fragments of cDNA aligned to the reference genome. So they've basically been placed on the reference genome in the place where their sequence matches the best. And from this, you can basically look at two things. So one of those things is, as we said, the genome structure. And what you'll notice here is because we align these to the reference genome, which has the introns in place, the coverage looks like you see here. It's going to have blocks where you have reads aligned. Those represent coding sequence, sequence that is transcribed. So those are going to correspond to the exons. And then you'll also have blocks where there's no or very little coverage. And those are going to represent the introns. Those are not present in your mature mRNA, and therefore they will not be present in your in the pool of material that you sequence. And so when you align everything back, you end up with these gaps. And we'll go in a little bit more into how we can use those to predict gene structure later. The other thing you can look at is how many reads align. So the idea here is that if you have more of one transcript than another transcript in your mature mRNA, then after you've sequenced, you will see more reads from one transcript than the other transcript. In this case, we're only looking at one transcript, but you can see here that two of these exons have a higher level of coverage and one has a lower level of coverage. And from that, we can actually infer that this exon isn't always present. So we can infer that there's perhaps two gene models here, one with 3 exons and one with only 2 exons. And that would be why this particular exon here has lower coverage. This same idea is how we look at differential expression. So if you imagine these 2 exons are actually completely separate genes, we could then say that this gene is transcribed at a lower level than this gene. And again, we'll come into the details of that a little bit more later on. Okay, so for the purposes of today, we're going to assume that your RNA, you've taken your mature mRNA, you've turned it into double-stranded cDNA, it's been fragmented, and we're going to assume that we're going to sequence that with Illumina sequencing. So if I were doing this lecture in person, I would normally go through Illumina sequencing. It makes for a very long lecture. And so in this case, what I've done is I've put up this QR code. This goes to a YouTube video called Overview of Illumina the Illumina Sequencing by Synthesis workflow, and that video is hosted on Illumina's official channel. So that is Illumina's official video describing how Illumina sequencing works. If you already know how Illumina sequencing works, that's fine. If you don't know and you're curious, then it might be worth going to watch that video before you move on with the rest of this lecture, because it might help it make more sense. Okay. After you've done your Illumina sequencing, you end up with files called FASTQ files. If your sequencing is single end, you end up with 1 FASTQ file. If it's paired end, you end up with 2 FASTQ files. And each file contains all of the reads you've sequenced. For an RNA-Seq Illumina dataset, it would be typical to have tens of millions of reads, depending on the size of your organism's genome. And each read is encoded as a block of 4 lines in the file. So line 1 is a unique header. That header can contain, it must start with this @ symbol, and then it can contain anything you like, as long as it's unique to the read. This one has come directly from an Illumina machine, and it actually has information about the machine serial number. This is the flow cell. This is the lane. We've got the coordinates of where this read actually was on that flow cell and some information about the barcodes. But it's also perfectly fine just to have read 1, read 2, read 3, or whatever, as long as they are unique. Line 2 is the sequence of the read, so that's going to be your usual string of A's, C's, G's, and T's. Remember, Illumina reads are quite short, so you have lots of them, but each one is only going to be, depending on which version of Illumina you've used, a maximum of around about 350 bases, and more typically about 75 to 100 bases. Line 3 is just usually a plus sign. And line 4 contains this string of letters. Now, each letter represents a number. And the reason we use letters is because the numbers are 2-digit numbers. So actually, by encoding each 2-digit number as one single letter, we reduce the file size quite considerably. So that's the reason why you see this string of letters. If you want to understand how the numbers are turned into letters, go and have a look at this link here. That will give you an idea of how this works. Each number represents the quality of each base. There's one of these letters for each base in the sequence, and it represents the probability that the base call was incorrect. Or you can also think of this as the base call accuracy. So a quality score of 10, the scale here is called the Fred score. So a Fred quality score of 10 represents a 1 in 10 probability of an incorrect base call. 20 is a 1 in 100 probability of an incorrect base call. 30 is a 1 in 1,000 probability of an incorrect base call, and so on. So they're logarithmically linked to the probabilities. The take-home message here is that a quality above, the bigger the number of the quality, the better the quality, and a quality above 30 is considered good. Okay. So what do we do with these? Before we can use them, when you think about RNA-Seq, especially when you think about those differential expression questions, we're usually thinking about quantitative data. But what we have here is not quantitative. We've just got these strings of sequences and their associated qualities. So we need to do some processing before we can get to the quantitative part of the data. The first thing we usually do is some quality control, and a very common tool for doing this is FastQC. And FastQC's primary job is actually to go through these FastQ files and look at all of these strings of quality data, turn them into numbers, and then make a plot of them. And this plot is going to look like this. So on the x-axis, we have the length of the read. So this is the first base in the read, second base in the read, third base in the read, fourth base of the read, and so on, all the way up to the end of the read. The y-axis represents that quality score. And the boxes are basically representing the quality at that position over all of the reads you've sequenced. So what you can see in this one is that this is actually very typical of Illumina. At the beginning of the read, FastQC also helpfully color codes it. So green means, yeah, the quality is fine. Orange means it's okay, but not amazing. And red means it's basically not usable. So what you can see very typically of Illumina is that at the beginning of the read, the quality is generally good. And as you go through the read, the quality deteriorates. And that's due to a problem called phasing that I'm not going to go into, but it's just a sort of artifact of the way that Illumina sequencing works. So the primary job of FastQC is to give you this plot and let you know if your sequencing is actually usable or not. While it's doing that, FastQC also reads all of these sequence rows as well. And that enables it to generate some other plots for you to give you an idea of, just a vague idea of what might be going on in your data. The most useful one of these for me is the GC content plot. So with this, you can see if your GC content is, well, you can get an idea of what it is. And you can also look for things like bimodal peaks, which might indicate contamination in the data. If your data looks like this quality plot at the top, usually the first thing we want to actually do with it is trim it. And that's because these bases at the end where the quality is lower have a higher likelihood of being incorrect. And if they're incorrect, then it's going to affect how your reads map to the genome. And so what we're going to do is we're going to go through each read, and if the base at the end is below 20, we're going to chop it off. And if the next base is below 20, we're going to chop that off, and so on. And that happens for every read individually. There are many, many different tools that do this. The one I've named here is Sickle, but there are lots of different options for this. And as I said, Sickle's primary job is to remove poor quality reads from bases, from the 3' end of each read. If the remaining read is then too short, it's going to discard it. And if you have paired end sequencing is going to check that all of your reads are still paired. Some trimming tools can also remove adapters. So adapters happen if the piece of RNA or cDNA you're trying to sequence, which is represented here in red, is really short, then the piece of it that you're actually sequencing, your sequencing can actually go all the way through that. Oh, sorry. And then go into this adapter at the other end. And again, this adapter sequence should not align to your reference genome, so we can trim that off as well. The next thing we do with it is align our reads to a reference genome. I'm not going to go massively into the details of alignment. I think most people here should understand the concept of what an alignment is. In this case, we have one very long reference genome, and we have a large number of very short reads. And what we're basically trying to do is place each read on the reference genome in the place where it aligns best. And we usually use this kind of scoring system to pick the best possible alignment, because short reads can normally align in more than one place. With RNA-Seq data, there's a few extra caveats to think of here. So, and this is based on what we were talking about earlier, where the mature mRNA will have the introns removed, but you're aligning it to a reference genome where the introns are present. So this is our mature mRNA in our sample, and this one has 2 exons, which have been spliced together, a red exon and a blue exon. And when we make our library for sequencing, we're going to sample randomly from all the way across this mRNA. So some of these reads are going to be in the first exon. Some of these reads in blue are going to be in the second exon. But there's also, just by chance, going to be some reads that cross the exon boundary. So part of them is in the first exon and part of them is in the second exon. We're aligning this, remember, to a reference genome. So now these 2 exons have been split and there's an intron in between them. Oh, that's not a problem for these red reads or these blue reads. They can just be aligned where they should align. But for these split reads, it is a problem, because what the alignment tool needs to know is that it's okay to do this, to align part of the reads in one exon and part of the read in the other exon. It's actually really important we do this. This is essential for if you want to use your RNA-Seq for gene structure prediction. But it's also, you can lose a lot of signal if you don't do this, particularly if you're working in organisms with lots of very short exons, because you'll then find that the majority of the reads actually do span introns. Whoops. Okay. So the 2 main tools you'll see for mapping RNA-Seq data, and as I said, the criteria for this is that they must be capable of aligning these reads that span introns. HiSat2 is really fast. It's fine for most use cases, but it does lack a bit of sensitivity compared to STAR. STAR is very sensitive, so I might use this in an example where I'm expecting to have difficulty with the mapping. It's also commonly used for single-cell RNA-Seq, but it's much, much slower than HiSat2. For today's thing, you're going to be using data that's been aligned using HiSat2. Okay, so just to remind you of this again, we have our reads and we've aligned them to a genome. So there's a schematic of what this looks like here. If you load your alignment into JBrowse, you're going to come up with something that looks like this. And I apologize, these are genes that are actually not from a fungal organism. Please ignore that. It's just because I happen to have these slides from a different lecture. The concept is exactly the same in fungi as well. So here we have two samples, one at the top and one at the bottom. What we're looking at here is this is a gene model with 1, 2, 3, 4 exons and 3 introns between those exons. You can think of these, well, I was going to say you can think of these like plots. They actually are plots. The y-axis, the x-axis on this plot represents the location of the genome, and that's the same between this gene model and these two plots. The y-axis for these plots represents the number of reads that's aligned. So from this, you can see two things. Firstly, this bottom sample has more reads in the y-axis, like these peaks are taller than the top one. That's an indication that in this sample, this gene is expressed at a higher level than it is in this top sample, and we'll come back to that later. The other thing that you can see is that our gene model stops here, but the transcription does not stop here. The transcription carries on for quite some way after that, and that's quite consistent between these two samples. So this is the sort of data that is telling us about gene structure. If you look at this a bit more carefully, you can see that there's a lump of transcription here. There's a block of transcription here that matches this exon. There's a block of transcription here that matches this exon, and there's another block of transcription here. You'll see that this one at this end and this one at this end, the transcription on the plot does not actually match our gene model. It's likely that in this gene model, the start is probably correct and the stop codon is probably correct, but the UTRs are missing. And I would guess that this gene probably has both a 5' UTR, maybe starting out here, level with this, with where this one ends, this begins, and quite a long 3' UTR, which probably comes all the way out to here, level with where this block of transcription stops on this plot. This one's a fairly simple example, so I think this gene model itself is probably correct. It's just the UTRs that are missing. But sometimes you can find things that are just really wrong. And so this is an example of this. You can see the gene model at the top. It has a UTR in gray. It has a coding exon, it has an intron, it has another coding exon, and then it has a 3' UTR. But if you look at the transcription from the RNA-Seq, it doesn't match what we're seeing up here at all. You can see that there appears to be a gap, probably an intron, but you can see that that intron actually sits right over where this exon is in the gene model. You can see that there's no transcription at all at this 5' end, and you can see that the transcription at the 3' end extends far beyond where this gene model thinks it stops. So I would guess from this that this gene model here is just wrong, and that in reality we're probably going to start transcription, about here. There's an intron from here to here, and then transcription is going to go all the way up to here. And I'd have to look at the sequence to say where's the stop codon, so where does transcription, what's actually translated and what's going to be UTR. The other thing you can look at in JBrowse is this thing. This is our intron junction track in the box at the bottom. And these marks represent the reads that are aligned like this. So we already looked at this idea that we have to split the reads in the alignment. This mark represents the gap here. So the gap from where this red bit ends to where this blue bit begins. And the thicker and darker the line is, it means that there's more reads that stop and start again in exactly the same place. And that's additional evidence that there really is an intron in this location. Okay. So yeah, I would, as I said, I think this gene model is just wrong in lots of different ways. Another thing that you can do if your sequencing is stranded is look to see if you're seeing transcription from the correct strand or the opposite strand. And this is another example where we have a gene model with 1, 2, 3, 4 coding exons and a UTR on each end. And here we've got stranded RNA-Seq. So if the plot is blue, it means it matches the direction of the gene model. And if the plot is red, it means it's actually transcribed antisense the gene model. So in this case, if we sequence females, we can see that the transcription matches the gene model fairly well. Maybe there's a little extra 5' UTR, that's a bit unclear, but the exons appear to be mostly in the right place. The UTR appears to be mostly in the right place. There's a small amount of reverse transcription or transcription from the antisense strand as well, but that's kind of minimal. If we look at the male, we find that it seems that only part of this forward strand transcript is being transcribed. There's no transcription at the left, the 5' end of this, but the 3' end looks fairly normal. But at the 5' end of this transcript, we seem to have a antisense transcript appearing instead, and that's much more prominent than it is in the female line. Again, we have evidence from the split reads showing that these exons appear to be correct on the forward strand, but there are also some highly supported exons on the negative strand as well. Okay. So that's some thoughts on how we can use RNA-Seq to look at gene prediction, look at gene structure, either predict new genes or check that the gene models you have are correct. I suspect most people are more interested in quantification of expression. So let's go back to this original plot. We already talked about these UTRs, but we also talked about the idea that on the y-axis, which represents the number of reads mapped in each location, you can see that these two are both on the same scale. And so you can see that the bottom one has more reads in the y-axis than the top one. So the basic inference from that is that the bottom read this transcript was expressed more in the bottom sample compared to the top sample. But when you've just got two samples like this, it's difficult to know if this is real. So to assess this, what we want to do is statistics. And to do statistics, we need quantitative data. So how do we get from this to numbers that we can actually do statistics with? Okay, so yeah, we need to turn our alignments into numbers. The other thing we're going to need to do here is replicates as well. Replicates, if you're doing statistics, is something you always need anyway. But we really want to see that this difference is consistent between these two conditions and not just something random that happened. Okay. So the very basic way of quantifying our data is to simply count how many reads align to each gene. That sounds straightforward, but the reality is that, as we just saw, the gene models are not always correct, and you can get some weird things happening in the alignment. So the simple case is we have a gene and the read sits purely inside that gene. But you can also get reads that overhang the end. For example, that is probably happening here. You can maybe get reads that go across the intron. That may be if you've managed to get some immature RNA in your thing, or if you have in your sample, or if you have some intron read-through mutations. You can get your split reads that look like this. You can get reads that overlap two genes because the genes themselves overlap. or you can get reads that could actually map to more than one gene. And as you're counting, you need to decide what to do in all of these different cases. This diagram comes from the documentation for HT-seq count, and with HT-seq count, it's common to choose the union option. So basically, if a read can only be aligned to one gene, it will be counted. So everywhere it says gene A, it's counted, even if it's not perfect, so if it's overhanging or over the intron. But if it's ambiguous in any way, so if it overlaps two genes, either because the genes overlap or because the sequence is very similar, then it won't be counted. That means that you're very sure that the reads that you have counted are in the right place. But if you have, for example, multi-gene families where the reads could map to multiple members of the family, you're going to lose out on those reads. And in those cases, you may need to consider different options. There are lots of different tools that do read counting. The diagram comes from HT-seq count because it's quite a nice diagram, but the one that we've used in the workflow, the one that you'll get data from today, is something called string tie, the one at the bottom. And I've put some other options here if you want to look at them. We can't use the raw counts, so we can't just count the data and use those counts directly. This plot is from a real experiment. And this is a pathogen that, when it infects its host, it replicates in the blood. So there's lots of it in the blood. The blood's in green here. It can also sequester itself in the brain and in the skin. But the pathogen burden in those two locations is much, much lower. So when you do your sequencing and you count how many reads have mapped to the pathogen from the blood, you get lots. And if you count how many reads map pathogen from the brain or the skin, you get much fewer. Now, if I were to do a differential expression at this point with this data, this is just the raw data. If I were to say, okay, what's upregulated in the blood compared to the brain, the answer I would get would be everything. Now, that's not actually true. It's simply that there are more pathogens in the blood than in the brain. So if I want to ask actually what's expressed at a higher level in those pathogens, I need to normalize this data. And what I'm basically trying to do is to bring these box plots into line with each other so that I can make comparisons within them more accurately. Okay. So there are two things we need to do with the normalization. One is just what I was just talking about, scaling units for comparison between samples. So here we've got a sample where we had a lot of RNA. This could be our blood sample, for example. And we have a sample where we have a low amount of RNA. This could be our brain sample. And so if we look at the same gene in each of those samples, it's very likely that we're going to get more reads aligning to the gene from sample 1 than we are to the gene in sample 2. And that is most likely to be simply a factor of the idea that we've got more RNA in total for sample 1 than we do for sample 2. So if we were to do a comparison directly, it would look like this gene is upregulated in sample 1, but we can account for this by scaling to the total read count. So what we're basically doing is we're saying, if we pretend that both of these samples have the same number of reads, what would the data actually look like? And then you can end up with a more accurate comparison, as we're seeing over here. If you want to do any comparisons within your sample, there are extra things you need to do. And the idea here is we're looking, within one sample here, and we want to ask, is gene A upregulated compared to gene B? But the problem here is that gene A has 3 exons, and one of those exons is quite long, and gene B only has 1 exon. So although you can see here that the coverage level is roughly the same, if I were to count all the reads that map to gene A, I'm likely to get a larger number than I am if I count all the reads that map to gene B. And so to account for that, we need to also scale for the gene length. And conceptually, the way we do this is we take a kilobase chunk, or we normalize it to the length of the read in kilobases. And you can see here, if I've put a hypothetical kilobase chunk on this gene with the red tracks and on this gene, the number of reads within that chunk is going to be roughly the same. So that takes us from this thing where gene A looks like it has more reads to, again, a more sensible comparison. So the way we do this is something called TPM, or transcripts per kilobase million. And that is a normalization method that is accounting for both of those two things, both the between sample normalization and the within sample normalization. So the way we do this is for each gene, we're going to count the total number of reads, and we're going to divide that count by the length of the gene in kilobases. And that part is doing our within sample normalization. That gives us a value that we call the reads per kilobase, or RPK. We then count up all of the RPK values for all of the genes in our sample, and we divide that by 1,000,000. And that gives us a scaling factor, and that scaling factor is accounting for the between sample normalization. And then we're going to divide for every individual gene. We're going to take the RPK that we calculated from that gene in step 1, and we're going to divide it by the scaling factor that we calculated in step 2. And that's going to give us our TPM. In the data you've been given, we counted the reads with string tie, and a string tie will also give us the TPM value. So we get both of those things from string tie. Okay, so this was our data before normalization, and after normalization, you can see that everything is looking much more aligned, and now I can make genuine comparisons between the blood and the other tissues here. Once you've done this, what you basically have is for every gene in your organism, you have a value for every sample you've sequenced. And at that point, your data is quantitative data. And you can do anything with it that you could do with any other form of quantitative data. So I could look at the expression of an individual gene. This data comes from an inducible RNAi knockdown experiment. So there were 2 cell lines. This is the 2 different cell lines before they were induced, and this is after they were induced, and the gene is the one that was being knocked down. So this was just confirming that the expression of this gene did decrease after we did the knockdown. You can do statistics. So it's usual to do pairwise statistical tests. That's what differential expression is. It's basically doing a statistical test for every gene in the genome to say, is it statistically different between my 2 conditions or not? It's very common to display this as volcano plot, as shown here. So with this, the higher up it is on the y-axis, the more significant it is, and the further out it is on the x-axis, the bigger the change. So this allows you to visualize both the magnitude of the change and the significance. Yeah. And you can also do advanced analysis depending on what your experimental setup was. So this one, this was a part of a clustering analysis where I've looked at expression of genes over a time course, and I've put them into groups where all the genes in the group behave in roughly the same way. So these all decrease towards the middle of the time course and then increase towards the end of the time course. And you can see I've plotted this in 2 different ways, both as the line plot here where the grey bars are the individual genes, and the red is an average, and also as a heat map, so you can see what's happening with every separate gene as well. Okay, so for the exercises tomorrow, you're not going to do all the processing yourself on this occasion. That has been pre-done for you, and we have done that using a workflow in Galaxy. So Galaxy, most of these tools that I've talked about are designed to be run at the command line. Galaxy is a web server that has these tools installed, and so you can upload your data to the server, choose which tools you want to run, and then run them using a sort of point and click interface that you might be more familiar with. And you can chain them together into a workflow. So this is the workflow that has been run for you to generate the data you'll be using. The input was reads, so these are FASTQ files, and these had already been trimmed. So you'll see there's no trimming step in this, that's already been done. And we also need the reference genome. Because the reads had already been trimmed, we go straight into the alignment. And this is the HiSat2 aligner, which, if you remember, is a splice-aware alignment tool. After that, we're going to do two things. One of which is we're just going to convert the output of HiSat2 to a different format. And that is so that we can use it to visualize in JBrowse. So we can make those same kinds of plots that I've been showing you by uploading that file into JBrowse. The other thing we're going to do is run StringTie, and StringTie is doing two things for us here. It's going to do the raw counting, and then it's also going to do the TPM normalization. To do that, we need to give it the genome annotation, and that's a file that just has the coordinates of every gene in the genome so that the counting tool knows where each gene is. And then the final thing we're doing here is the output from StringTie has lots of columns in it, but a lot of them are irrelevant. And this last tool is just extracting specifically the columns that we need for upload to FungiDB. So if you want to look at this a bit more, you can that there are lots of links in the exercise to this workflow in Galaxy. You can go and have a look at it. You can import it into your own workspace. It's free to set one up and you can use it. If anyone has their own data and wants to try this, then please let us know. I can help you with this. Stuart is also a good person to help you with this because he has lots of experience with Galaxy. Okay. The first exercise on Thursday is using the My Datasets tool in FungiDB. And what My Datasets does, or one of the things that My Datasets does, is it allows you to run a workflow that looks like this, and then upload the output from that workflow into FungiDB. So you can upload these files for JBrowse, and you can upload this file here. And that allows you to use a lot of the tools that you've already explored in FungiDB to explore your own data. So you can view your data in JBrowse. You can run fold change queries the same way as you did at the beginning of the week on preloaded data. So that's the JBrowse plot there. You can view plots of your data like this. Those will be generated for you automatically. So the first exercise on Thursday is understanding how to do that and how to access data if you've uploaded it. And if any of you have your own data and want to try and get an RNA-Seq workflow running so you can look at your own, please let us know. The other exercise on Thursday is about exploring gene models, and this is this concept that I talked about here. We have some examples for you in fungi where the gene models and the RNA-Seq and potentially some other data like ChIP-seq as well don't match each other. And you'll have the opportunity to explore some of these and see if you can interpret and come up with some idea about what the genes actually look like based on the functional data. Okay, so that is the end. I hope you enjoyed the two exercises. If there are any questions, please do grab me and ask. And thank you very much.